This is Jane Doe, otherwise known as my Gene2 patient 1930. Now, Jane seemed like a perfectly normal, healthy child for the first many months of her life. But when she began walking, doctors described her walk as gait ataxia. Around the same time, Jane started having seizures, then developed pancreatitis, and because of all of this, rapidly started losing weight. So later genetic testing revealed that Jane has de novo mutations in the genes SSPO and NBEA. Now, the curious thing about Jane's case is that if you look in resources like OMIM and Orphanet, there's actually no connections between these genes and any of these phenotypes. So Jane has what we call a rare and undiagnosed disease. Now, paradoxically, rare diseases as a whole are not that rare. It's estimated that somewhere between 25 and 30 million Americans suffer from a rare disease. That's about 1 in 10 people. But despite not knowing any direct connections between Jane's genes and phenotypes, that's not to say we don't know anything about the genes and phenotypes themselves. In fact, thanks to the vast number of biological ontologies, we actually know quite a bit about them. We know how the phenotypes are related to one another in the human phenotype ontology, and we know how these genes or proteins interact with each other based on the string protein-protein interaction network. Now looking at Jane's genes and phenotypes in the context of these larger ontologies, and then overlaying the known connections between OMIM and Orphanet, you can see that there are potential explanations for Jane's condition just through indirect paths. And what I'm interested in doing in this work is identifying regions of a graph like this that are dense and indirect connections that potentially answer cases like Jane Doe. And the way that I've decided to go about doing this is using network clustering methods. So a clustering algorithm, for example, might identify 3 clusters within this little toy example. It might find this orange cluster within HPO, maybe this green cluster within string, but in the case of somebody like Jane Doe, what we really care about would be something like this gold cluster in the middle, something that spans HPO and string and groups together terms related to a single patient like Jane Doe here. So this is exactly what we're doing and what I'll be describing here. We're going to create a heterogeneous network of genes and phenotypes, cluster it, and then identify clusters that we think will be useful in the future for hypothesis generation and further investigation. So this is just to point out the scale of the network we're going to be working with, that in the end, the network has around 35,000 nodes and 6 million edges. So the network is constructed by combining HPO and string using annotations from OMIM and Orphanet. Fortunately, these annotations are made available by the human phenotype ontology. So I construct what this network would have looked like in the years 2021, 2020, and 2019. The previous versions of this graph are going to be useful later on in the development and evaluation of the method. Next, we cluster this network. So I use 4 different network clustering algorithms here: greedy modularity, walk trap, infomap, and SESNA, which is a label propagation method. Now these represent like 4 very different classes of algorithms to evaluate how these methods work in general. But using just these 4 algorithms, they have a tendency to produce relatively few clusters that are occasionally really large in size. So if you look at the distribution of the size of these clusters here, you can see that some of them are huge. Like Infomap has 1 cluster that has 25,000 members in it. Now, if you were to take a list of 25,000 genes and phenotypes and present it to a doctor as containing the solution to somebody like Jane Doe, you're going to get laughed out the door. That's way too much information to be useful. So as another study noted, in order to be useful for human manual curation, a cluster needs to have no more than 100 members. So to reach this threshold, we introduce another layer of clustering using the Paris hierarchical method. The nice thing about Paris is it allows you to set an upper bound for cluster size. So we cap our cluster size at 100 and then recluster our previously identified clusters. This results in, first of all, a lot more clusters, as you can see, but all within this manageable size, ranging from somewhere between 2 members to 100 members. So we're now to the state that we have a lot of clusters that are potentially manageable, but we need some way to score them and understand how useful they are. So the way we answer this question is referring back to those previous versions of the network. So we start by clustering the 2019 graph. So we'll use two toy examples here of the green cluster and the gold cluster. And then we identify the edges that were added in 2020 to the network as a whole. So these two examples up at the top represent future gene-to-phenotype connections that were added the previous year. So then what we're going to look for is where the two nodes of one of these future edges co-occur in one of the 2019 clusters. And if they do, we're going to call this a rediscovery. And then, so that's a good match. But then in the case that something like the second example occurs, where the future edge is split between two clusters, that's a miss, a non-useful piece of information. So we generate a list of all of these future edges and use it to score all of our 2019 clusters. And this is the result we get. So we create a null model using a snowball sampling method to create 10,000 iterations of each cluster, which allows us to create an expectation for the number of rediscoveries. We're then doing a log2 ratio of the number of rediscoveries we observe for each cluster versus how many we expect. So using this little equation, if there are an equal number of observed rediscoveries and expected rediscoveries, this would be 0. And you can see that in all of these graphs, there's a spike right at 0. So the vast majority of all of these clusters we identify have as many rediscoveries as you'd expect based on our null model. Then there's a small portion that have a negative value here that's indicative of having fewer rediscoveries than you'd expect. And then there's the small handful, somewhere between 7 and 19%, that contain more rediscoveries than you'd expect at random, typically greatly exceeding the expectation. So we need some way of prioritizing the 7 to 19% and isolating them ahead of time that we can hand off to physicians for further investigation. The way that we decide to go about doing this is using an XGBoost model. So we trained this machine learning model on some biological features. These would be things like tissue, disease, or GO enrichment, and then also some cluster topology measurements, such as the internal degree ratio, the number of triangles, or the ratio of genes to HPO terms within a given cluster. So in actuality, in our 2019 data, there are 1,898 clusters that have a p-value based on our snowball sampling null model with a p-value less than 0.0001. That is to say that the actual observed cluster has more rediscoveries than all 10,000 of the simulations we performed. So if we pull that same number of clusters from the list predicted to have the highest score to be the most significant by our XGBoost model, we find that our actual most significant clusters and those predicted to be the most significant overlap by about 71%, or that there's 1,351 clusters in common between these two groups. So we're now at the state that we've created a heterogeneous network. We can cluster it into manageable size groups. And now we can relatively accurately predict which ones contain future information, which we can then hand off to a clinician for further investigation. We then applied this method to Jane Doe and the rest of the MyGene2 patients. In the case of Jane, we found that her gene NBEA and the term for weight loss co-occur in one of our walk trap communities. Now, the interesting thing here is that after we had this result, I was reviewing Jane Doe's information on the MyGene2 website and found she was cross-listed in the Undiagnosed Disease Network out of Harvard Medical School. In the second profile, it had been updated much more recently than MyGene2, and the researcher that updated noted that they'd experimentally validated and solved Jane Doe's case and had also identified it as being the gene NBEA that was causing all of her phenotypes. But Jane is just one of 912 undiagnosed patients on the MyGene2 website. Of these, today, 115 still remain unsolved. So of these patients' genes and phenotypes listed on MyGene2, there are no connections between any of them for 115 people. But of these, 13 are explained by one of our clusters. So this is 13 families that have gone years in a diagnostic odyssey that now have a new potential answer at hand. So based on our preliminary success, we have an ongoing collaboration with Children's Hospital of Colorado and the Precision Diagnostics team. And we hope to implement this and further evaluate it on the patients coming through their hospital every week. And now I'd like to thank my co-authors, lab mates, and the various people that have been helpful with this project along the way. And I'll take any questions now.
Great presentation. I will try to see if you can find any relations from model organism databases instead of just human.
No, I haven't, but I was actually speaking to somebody earlier today that explained an ontology to me relating those two things that I'm actually really excited to use in this and a couple other works going forward.
We have to talk again.
Yes.
Hi, it's Melissa Handel. That might be the ontology development of my group. Great to see you give this talk. So a couple questions. So one is that I love the sort of predictive work that you've done and sort of taking the different versions of the ontologies. As an ontology developer, we often go in fits and starts, and so parts of the graph get really fleshed out, and then that part gets left alone for a long time, and then we go somewhere else. And so that's my first question is, how do you sort of build that kind of overall topology into the model, recognizing that human beings are actually working on different parts and at different rates? And then the second question is just understanding how you're reconciling OMIM and ORFANET. We spent a lot of time doing that as well, and just trying to think about how you might leverage some of those mappings and reconciliations, or if that's something that you've already done, or is that to do?
Yeah, so about the OMIM and ORFANET thing, we're actually not doing any of the reconciling. We're taking advantage of the work done by the people at the Human Phenotype Ontology and just using the annotation off of their website. Saves us a lot of legwork. And then to respond to your first question, this is actually something we're actively looking into. Earlier this year, I actually submitted a grant to look into that bias and what's really causing that unequal growth within the ontologies. So we can look into that and how that might be biasing our methods and other people's in systems biology.